Welcome to another episode of Times and Tides. Today, we are going to now discuss with the two accomplished scientists on a question that perhaps has been haunting philosophers and great thinkers almost for several thousands of years. Maybe ever since man began to now question his own existence. The question in particular is, who are we? But let me place a caveat in advance that we are not going to discuss the philosophical side of this question of who we are. Now, who are we or who are you is being now questioned here from a scientific perspective. Because as you will see as we go through the podcast, developments in the last 2 or 3 decades in the area of microbial biodiversity, human health, and the relation between microbial diversity and human health has shown that scientists also have begun to question, after all, who are we when we say we refer to ourselves. In that sense, this question of who are we, which is perhaps falling in the junction between philosophy and science, is also being prompted by the developments of science, and that part of the science we are going to discuss today. To discuss on that, we have two experts with us. First of all, let me welcome Dr. Yogesh Shouche. Yogesh, welcome to this show. I will talk about what is the contribution of Yogesh a little later, but let me also welcome Dr. Chirag for this episode. I know Dr. Yogesh Shouche for the past maybe 2 decades. I should say that I was prompted by his talks both in Department of Science and Technology in New Delhi and one specific talk he gave in Kargil when we had a workshop to train the young college students. Dr. Yogesh Shouche worked in Pune as a microbial ecologist, especially in the National Center for Cell Science. Being there, he developed his interest in not only microbial ecology but also in collecting the diversity of microbes across the country and studying their futures. Prompted by his commitment and work, Department of Biotechnology and Government of India suggested him that he expand his work from merely studying diversity of microbes to also expand his work towards how these microbes are also perhaps involved in the human health. So he extended his work to collect microbes from across the country from the human gut and human body also. I must add here that one of his contributions to the country is what is called as MCC, Microbial Culture Collection. This microbial culture collection that he has maintained in Pune in National Center for Cell Science has become a globally well-known center. The International Depository Authority and World Federation of the Microbial Cultures have recognized this, and beyond that, he has been a frequent publisher because of the quality of science he has been doing. He has more than 400 publications. Am I correct, Yogesh?
Yes, yeah.
He has been recognized by major science academies of the country, National Academy of Sciences and Indian National Science Academy, Maharashtra Academy of Sciences. Perhaps you will appreciate the contribution done by Dr. Shouche if I tell you that one among the most important genus of bacteria is named after him. In fact, belonging to this genus is the Bacillus clausii, which you might have taken if you have taken prebiotics. So you have consumed a bacteria that is named after Yogesh Shouche, and that is the kind of contribution he has got. Right now he is in Bangalore. He is now, of course, after leaving Pune, he joined Ajim Premji University, and now you are in
Scan Research
Institute, an independent organization. Yeah. We are very happy to have you here, Yogesh. Thank you very much for taking your time. We also have a doctor with us, a neurosurgeon. I'm in fact very happy to introduce neurosurgeon because this particular topic that we are going to discuss has so much to do with the neurological behaviors of the humans and also the other stress or psychological events of moods of human beings. Dr. Chirag did his MBBS in Bangalore Medical College, and after that he went for a long training and also postgraduate course in neurosurgery in Nimhans. All of course, the entire world knows what Nimhans is, and he is now a practicing surgeon. But beyond that, his interest is in maintaining fitness of or health fitness, and just not restricting his own to his own field of neurosurgery. He has been now working and collecting and studying the area of this human biome or human microbiome, and the reason why he is here is because of his personal interest in expanding his own knowledge and also in studying the possible application of the field we are going to study for his surgical and his clinical studies. There will be a lot of differences of opinion between the scientist and the medical doctor, obviously, because the scientist is always enthusiastic in applying his field, whereas the medical doctor is always now very careful about now introducing what we now discover as new discovery into field. You perhaps will enjoy listening to this, these differences of on the issues of application of science that Yogesh has found versus the application in Chirag. Welcome again, Dr. Chirag.
Thank you.
What I will suggest is perhaps now introduce to the viewers the topic in particular. I started telling that we are going to discuss on who are we. I will start with you, Dr. Yogesh. Why did this question came to you, scientists?
Yeah, so this is a very interesting question, and the question came because certain developments in science that happened in since the beginning of this century, they told us that all the ecosystems, not just human body, but all the ecosystems harbor a huge unknown diversity of microbes. What I mean is that we always knew that they are there, but we didn't know the extent of their diversity. That is because of, just to tell you briefly, because of the developments in DNA sequencing technology, that we could understand the microbes which we would otherwise did not know about, because we could not see the microscope, that microbe under the microscope, or grow that in the laboratory, but we can know about the microbe from the DNA, and that's how the DNA sequencing technologies helped us to understand more about these microbes. And like many other ecosystems, we discovered that we hardly know anything about microbial composition of our body, and then that led to the observation that the number of microbial cells in our body exceeds the human cells. So earlier it was thought that there are 10 times more microbial cells, but now more modest estimate is little more than our own cells in the body. So human body contains something like 30 trillion cells, human cells, and 38 trillion microbial cells. That is the current estimate. But still, if you look at the diversity of genome,
just can I stop here?
Sorry.
Your numbers, even if it is slightly extra, is a surprising thing to a man who is not introduced into that. So what you are telling is the microbial cells in our body is several folds, or at least more than our own cells.
Our own cells, right?
So that is the reason why the question is, who are we?
Huh? Yeah. So I was going to expand on this little more. That so if you just look at the number of cells, the microbial cells are little more than the total number of human cells in our body. But if you look at the genetic contribution, so the number of genes contributed by microbes to our body, so typically human body, in human genome we have 23,000 functional genes. As against that, if you count the number of genes contributed by all the microbes in our body, they are 500 times more genes are contributed by these microbes. And that's why the question is who we are. Because since the microbes reside in our body on all the parts, they are not lying there just silent. By their activities, they kind of affect all the activities of human body. So, and that's the reason there is this question as to who we are.
Again, a clarification. Again, an exciting point that you are telling. That is the total number of genes that these microbiota that are there in our body contributing to the, to the whole functioning is 500 times more than the genes that we ourselves are contributing to our survival.
Right.
Did I get you correctly?
Yes. The genes that are contributed by all the microbes in all different parts of our body is 500 times more than our own genes.
So obviously I'm only one, and whereas the microbes are 500 times.
500 times.
So the question, who am I?
Who we are. Right. Yeah.
Dr. Chirag, when people say that these genes of microbes are more than us, where are these organisms located?
So the microbiome can, or whatever these bacteria and other organisms that are not us, not our cells. So they can grow anywhere in the body which is accessible to whatever is external to us or whatever is outside us. So they can grow on the skin, they can grow on any of the surfaces where external sources can sort of enter our body. So they can be in the mouth, they can be anywhere within the gut, usually in the small intestine or the large intestine. They can be in the reproductive surfaces because they're also exposed to the outside world.
Okay.
So usually you have a skin microbiome, you have an oral microbiome, you have a gut microbiome. So all of these places have bacteria and also non-bacteria, other organisms apart from bacteria that are growing there.
Like for example, other than bacteria means fungi
You
and
can have fungi as well.
Yeah.
Okay.
You have fungi, you have unicellular eukaryotes, you have viruses.
Okay.
So all the four microbes.
Okay. So where are all these? I mean, you told on skin and gut and intestine and all that. Where is high number of microorganisms among?
The largest number is in the gut. So that is where the majority. So for example, 1 kg of your body is just your gut microbiome.
So year afterwards, I have to reduce 1 kg from my
I would not do that. You want that microbiome.
Yeah, you want that.
Oh, I agree, and that's why the question: who are we? So we are only when those organisms are there, and so that 1 kg contributes more than this remaining 65 kg of my weight in terms of some genes. Okay.
Absolutely.
Okay.
Yeah. No, and I would like to add something more interesting.
Please.
See, it has kind of shaken our previous concepts that we had, because for a long time we thought that breast milk is sterile and it does not contain any microbes. But then the research done in last decade or so has shown that breast milk not only carries a defined microbial composition, there are some 100 and odd different species of bacteria present in the breast milk, but these are the bacteria that eventually go to baby's gut and reside there and responsible for healthy development in the baby's gut. Further than that, there are these what we call as complex oligosaccharides. So these oligosaccharides that are present in mother's milk, they are not meant for baby. They are not meant for baby as a food, but they are meant for those bacteria as a food, so that good bacteria can grow fast in baby's gut.
Correct me. What you're telling is mother provides those microorganisms through the milk to the baby.
Right.
And also provides oligosaccharides through the milk to the baby, so that the microorganisms.
So that good bacteria can grow.
Oh, I see. So there is a perfect evolution of this.
Right. Yeah. And interestingly, again, I mean, we don't have time to discuss that.
No.
Yeah. If you see this in the evolutionary course, you will see that it is all across. It is not only in human. You would find such examples in insects. You would find examples even in a unicellular paramecium. You see there is an endosymbiote bacterium which distributed in 2 paramecium cells when the paramecium divides. So this microbe and
I was about to come to that. When you told that mother's milk contains the bacteria and the nutrients also to those bacteria, so that the whole package is going into the baby's body where they grow and help the baby. That means there is a kind of evolution not only in the organisms to pass on from generation to generation, but also in the host genome, that is the host body mother, to provide nutrients to that.
Right.
The whole complex has evolved as a positive.
Yes.
Wow.
I mean, in insects, you will find many interesting examples. Insects, for example, there is an example in insect where I don't remember the species, but when the insect lays eggs, it also, in one case, the mother smears the surface of the egg with her fecal matter, and when the larvae emerge from the egg, they go and lick that surface
I
so
see.
that they get the inoculum. In another case, mother deposits a specific capsule containing bacteria, and when the insect emerge, they pierce that and get the inoculum.
Okay.
So
I am from the University of Agricultural Sciences, as you know. There is a department of entomology there, and one of my friend is working on dung beetles.
yeah.
He did tell me that these dung beetles roll the dung from the cow dung, and when it puts there along with the egg, it also puts a small mass of its gut there,
Yeah.
which is supposed to be now having microbes.
Correct.
So, in other words, even before our mothers or our ladies provide the organisms and the milk, there is in insects providing the same. That means evolutionarily there has been a co-evolution between the host and the microorganisms. Correct?
Right, and mechanism to ensure that these microorganisms are transmitted from one generation to next
Next
generation.
generation, which are useful
Yeah.
microorganisms. I would like to ask you, Chirag. So, one example you told of microorganisms going from mother through the milk. How do we get this gut microorganisms through the milk or any other way? The baby, how does it get the microorganisms?
So the way that we start acquiring this microbiome, it actually happens within the period of gestation itself. So when you are a fetus, that time itself it begins a little bit. So when you're in the womb, you can acquire some microorganisms through the mother. And
But how?
so it will pass through the placenta, it will come through the blood.
I see.
And another way that it can happen is that when you're being delivered through a normal delivery, for example, not a cesarean section, you're coming out through the normal reproductive organs. So when you're passing through the mother's vagina, whatever bacteria is growing in the secretions over there, that can coat the skin of the child. And so that is one way that the child can acquire microorganisms. So what we have seen and what is very clear in research is that children who come out through a cesarean section definitely have different microbiome as compared to children who are born through a vaginal delivery. And it has also been shown that can be linked to certain diseases or certain abnormal or certain deviations from normal.
Meaning the babies born normally are more healthy, whereas the babies born through cesarean may have some problem. That's what you're. Then why do people go for cesarean for?
At the end of the day, it's not. Nobody is saying cesarean sections are bad. They are medically indicated for certain reasons.
But there are differences.
But there are differences when it comes to the microbiome. And for children who come out through a normal vaginal delivery, they will have a more diverse microbiome. So there, it's important for us to recognize and sort of understand what is a healthy microbiome. We all have microbiomes at the end of the day. Now, for example, there is no universal definition of what is a healthy microbiome. It's not like if I have this microbiome and I'm completely healthy and disease-free, that everybody in the world should have this exact same microbiome. It does not work that way.
I was about to ask this. When you both of you tell that so much of microorganisms are passing on from parents to the offspring, especially mother to the offspring, either through the milk or through the birth process, or it is. These microorganisms were generally, for a common man like me, were being viewed as dangerous. You should get them off, wash your hands. I mean, this is the kind of training given to generally public. So now you are telling, no, there are more useful organisms that pass through. Am I correctly getting?
Yes. I mean, if you look at the numbers, even and just talk about the human pathogens, today the total number of known bacterial species coming from all the sources is few thousand. And amongst that, if you want to list out not just human pathogen, human, animal and plant pathogen, the number would not be more than 1,000. The total number of and actual existing bacterial species is today the number will be something more than 20-25,000. So a small fraction is actually pathogenic.
Can I now tell to the viewers on another point of importance here? I think viewers may know that Dr. Yogesh collections at Pune has crossed 1.5 lakh microorganisms. So the total microorganism species is around 25,000.
Around because see, it keeps on increasing every day.
It's okay, known at least.
Yeah, total list is
among
around 20,000.
Those, the harmful are only 1,000.
You take plant, animal, human, everything put together. Only human will be just few hundred.
We are blessed with more of good than bad, right? But it's only with the bad we now always suffer and identify the suffering. There is so much of good that these organisms are doing. So we better not wash our hands carefully.
Not
I'm just joking.
that is another extreme.
Yeah, I think maybe I should ask this question to him. So should I not wash my hands then?
You should definitely wash your hands.
Okay.
But again, so
No, we should not give a wrong impression to the viewers.
Yeah,
That's
absolutely.
why. Yeah.
But one thing I would like to bring up here is when it comes to nature, every organism wants to survive.
Correct.
And so, for example, when we discuss pathogenic organisms or pathogenic bacteria, that's a slight bit of a flaw in the sense that these organisms are not doing their best to survive.
Yeah.
Let's say when you're talking about good bacteria or commensal bacteria, as we call them, they are living with the organism that they're inhabiting, right?
So, they have learned to live with the
organism.
organism.
And that is the best thing for them.
Achha.
Because they are growing, the organism is living, and they can continue to proliferate.
Achha.
Now, let's say that you have a pathogenic organism, it infects the body, it grows, and then it kills that organism. And then it is also dead, unless it is able to pass on to another organism or another body.
Okay.
So that is a bit of a flaw in the sense that living with the body is better.
I follow what you're telling. As a layman, let me now interpret what I got from you. So there are good organisms which are very good and they know how to survive, and they have been very beneficial to us. There are foolish organisms which do not know how to live, and they are causing problems to us. Yes. Can I interpret that way? I know it's a very wrong way of interpreting, but
it is an okay way of interpreting.
Okay, fine.
Yeah,
you have.
There is another side of it. For some organisms, definition of good and bad is contextual.
Achha?
Contextual in the sense that, for example, there is an organism in our gut called Akkermansia. So, under certain condition, it's a good bacteria, but under certain condition, it degrades your intestinal lining, and then that causes certain problems. So, in some cases, it is a contextual definition, good or bad. It's unlike classical pathogenic organisms.
Okay.
Here, the definitions are little.
Let me come back to this co-evolution that you're talking. So these organisms have learned to live with us.
Yeah. And we have learned to live with them.
We have to learn to live with them. So it does appear that if they have taken our body as their niche, as their habitat, they also would better help us in several ways. Just not sit there, but help us. What are the ways in which they perhaps are helping us? Example.
I mean, there is a long list of ways in which they can help
example.
us. But the classical, when we started understanding microbiology 300 years back, the classical understanding that time was they produce some vitamins and amino acids, for example, vitamin B12 group of vitamins and certain other vitamins which we cannot make ourselves. So we depend on bacteria to produce those vitamins in our intestine, and then those are absorbed and we go get those vitamins.
I have a question. Sorry if I'm interpreting, because that's because of my... Vitamin 12 or whatever it is, we can't prepare.
We can't make synthesis ourselves.
Then how did we evolve without preparing that? From the very evolution of human being.
It comes through our diet also, but the microbes supplement them.
Okay, so please go ahead. Sorry.
So that is one way. Second is another obvious thing is that there are lot of our food ingredients. For example, the complex polysaccharides coming from bananas or apples, like which is called as pectin. We ourselves cannot digest them. Left to ourselves, we can't digest. But the bacteria, these bacteria help to digest that. Then there are many other
things. Like the bovine organisms digest the cellulose.
Cellulose, correct. Okay. Yeah. So then there are other things like they train our immune system so that we are able to fight the infections. It's a kind of dummy war. So they
say it again.
They train our defense system of our body so that it can fight with any invading pathogen. So that was the classical understanding. But today we know that they do much more than that. So today from the same dietary fiber, they produce some small compounds which are called as short chain fatty acids. So these short chain fatty acids have a very central role in our metabolism, because first and foremost they are nutrient for our intestinal cells. So they help our intestinal lining to be healthy and properly developed, and they play a crucial role in regulating our metabolism, including the blood sugar levels. So and these are produced by our gut bacteria. And now we know, when we are talking about gut-brain axis, now we know that they produce many more compounds, like some neuropeptides, which effectively go and affect the brain. So these are some of the, and it goes more complex. I mean, I don't want to get into those complicacies, but it goes more complex because some of the compounds produced by these bacteria, they in turn change modify our host DNA in such a way that their expression is also changed. So it is much more complex.
You used a term, gut-brain
Axis.
axis. What is that? Can you explain that? What is gut-brain? I thought gut is independent and brain is separate. Why did you use that word, gut-brain? Chirag?
Yes, so the gut-brain axis is something that we have discovered over the last couple of decades. So basically, the body has the brain, the body has the gut, but the gut has its own brain itself. Right?
Say it again.
The gut has its own brain itself.
Sorry, not clear.
Okay, so for example, a lot of people think the brain controls everything. And all of the information has to come from the brain to control the rest of your body. But the gut, or the intestine, the stomach, all the different parts of the gut have their own nervous system as well. So there is some part of autonomy which is residing within the gut. So it is able to control its own function. Now, what is the gut-brain axis is a little bit beyond that. So the gut-brain is able to control its own functions with some material and some chemicals that it produces. A lot of the control is extended outside of the gut from the help of the microbiome, which also produces some materials. This will be like neurotransmitters, which are chemicals that the brain normally uses. Some short chain fatty acids, as Dr. Yogesh said, and some other materials that can travel from the gut and they can go everywhere within the body. They go to the endocrine system, they go to the immune system, and also, quite importantly, they go to the brain. So right from the beginning, right from your birth, they sort of influence how your brain is going to develop, and beyond the brain, the mind, how your cognition or your brain's capacity to think.
Please. So you told these microbes that are there in our gut, they produce some molecules, like short chain fatty acids,
and some other
they can
compounds,
travel so many places, also get into the brain and help the development of the brain. So to that extent, we are dependent on those microorganisms even for our brain development. And that is in some sense a gut-brain axis. Am I getting correctly?
Right, yeah.
You people are changing the whole biology that I understood.
We have not changed. Microbes have changed.
In a good way.
Okay, please go ahead. And so they help the development of the brain. But do they also now help the functioning of the brain?
They do. So these neurotransmitters, as we said previously, these are molecules that can help the brain sort of function in a normal way. So studies have shown us that gut microbiome and whatever it produces and the gut-brain axis affects your thinking capacity, as well as your memory, how you learn, your personality, your behavior. That is just how you behave as a normal, healthy adult. Beyond that, they affect diseases.
No wonder the question, who are we? They affect our memory. They may affect our thinking. Mood.
Your stress levels.
Stress levels.
Absolutely.
What are the studies that are done to now show this? Can you explain to the viewers in some simple terms the experiments perhaps that have been done to now show that these microbes are actually affecting our stress?
So usually how these studies are performed is we will take a group of people who do not have issues. They are called controls. And we will take a group of people who have these issues. They might have either, let's say, for example, autism, or they might have some other psychiatric disorders, or they might just have very simple non-disease issues, right? Just maybe mild depression, or they might have some different cognitive disorders, issues like this. So this will be our test group. And what we do is we take samples, or basically stool samples from both of these, and we evaluate what is the diversity of the microbiome in this, as to what sort of bacteria are growing within the people who do not have these issues, and what sort of bacteria are present in the people who have these issues. So what has been clearly shown by science is that the diversity can be different. That is, for example, let's say we can have anywhere between 300 to 3,000 species of microbes which are supposed to be growing within our gut. There can be more than that also and less than that also. Let's say that, for example, a very normal population with no problem has a much larger diversity.
Okay.
So studies show us that the people who have these certain diseases or even different non-disease issues
Okay.
have a slightly narrower diversity.
Okay.
They have
So let us say
lesser
the people
species.
with the depression will have narrow diversity of microbes in the fecal matter
Yes.
compared to a normal person.
Yes.
Okay. But that is diversity.
Yes.
But has it been possible to now ascribe that this particular absence of
Yes.
this particular microorganism is possible to
It
show?
is possible.
Acha. So they have shown.
They have shown.
For example, as you told, non-disease like mood problems or
mood,
depression.
stress, cognition.
Has any specific set of bacteria been shown to now be
Yeah,
responsible?
so that's a good question. I'll come back to that question in a while.
Please, yeah.
But before that, I just want to flag one issue. Since you, while passing, you made that statement, fecal bacteria. So the one important thing which is a kind of still limitation in these studies is that for these studies, all the studies today have taken fecal sample as a proxy to study the gut microbiome. And you can you'll imagine that the length of our GI tract, it's very long. What is the length of the GI tract total?
Up to 10 meters also.
10 meters. So you can imagine you are taking fecal sample as a representative of that GI
meters.
tract, and which is definitely not
Uniform.
the right uniform. It is not the right sample because it does not represent the entire diversity of GI tract. So that is a important caveat there.
Okay. I understand. You are telling that may not actually represent that.
Correct.
I find that.
Yeah. So and now coming to your question as to the changes, that is again a very interesting question, because when you say a change, there is normal and there is a change. So this change depends on what is your normal is.
Achha, okay.
And since the gut microbiome is influenced by variety of factors, including where you stay, what you eat, what is your lifestyle, so it's a very simple logic that across the globe, or even in a highly diverse country like India, we have 10 biogeographic zones and several what we call as several communities.
Yeah. I completely agree. But no, my question, let me come back. I'm sorry if I am holding you back. No, I completely agree. There are problems with this. But my curiosity was the following. Let us say I have 100 people who are normal. 100 people who are now affected by the mood disorders. Okay. Now I examine the fecal biodiversity. And as Chirag told, there is a less fecal diversity in general in the mood disordered people compared to healthy people. My question was, in addition to the reduced diversity, are there any specific organisms perhaps?
I was coming to that only. So the change of organism would depend on what is your healthy reference line is. So in different populations, you will find different organisms changed. And I can give you a classical example from our own studies. If you look at globally, and it's not about neurological disorders, but I'm talking about diabetes, because diabetes, obesity are more studied because the studies on diabetes and obesity started 20 years back.
Are you implying that obesity and diabetes is also linked to microbes?
They are also linked to
my God. First, let us talk about that.
Okay.
You mean obesity and diabetes are linked to microbes?
Yeah. So these are the classical studies which were done.
I'm sorry, we'll come to the organism little later, but this seems interesting.
Yeah. So when the microbiome science started, these were the classical studies. Probably diabetes came first because it affects a large population and people are always looking for some easy solution for diabetes. I mean, anybody would be happy to have a solution where there is no dietary restriction. You can eat whatever you want, take some tablet and eat whatever you want. So that is the interest. So then they discovered that in diabetic individuals, the microbiome of diabetic individuals is different from a healthy individuals, and today there are thousands of studies on that. I'll come back to solutions also later as we discuss microbe based solutions. So the point is these differences were found, but the problem is that when the studies started expanding globally, since the reference line in different populations is different, the change is also different. So
That's
the
okay. But there are
microbial taxa that
differences.
differ is different.
There are differences.
Yeah.
I have a question to Chirag on this. So let us say there is a diabetic person and a normal person. In the gut, there is a difference in the microbe. My question is, excuse me, I'm as a layman, I'm asking whether the difference is because there is a difference in diabetes, or is a diabetes because of the difference in the microorganisms? You know,
I'll answer that question first
Okay.
before it goes to
Okay, please.
I will also answer this.
Oh, please,
I
yeah.
may oppose, he will answer. But
Okay.
I'll answer the question
Yeah.
starting with a classical study which would kind of emphasize the microbial role in that.
Okay, please,
Because
yeah.
that was a study done in 2013, 12
Okay.
years back.
Okay.
And it is a very popular study where they took, in a pregnant woman, there is a condition called as gestational diabetes. So woman is normal in the first trimester of pregnancy, and in the third trimester of pregnancy, she develops diabetes. So they took the microbiome of the same woman in the first trimester, put it in the mice. Mice was normal. The same woman, when the same woman after she became diabetic in the third trimester, took the microbiome, put it in the mice, and the mice was became diabetic and fat. So that kind of strengthened the gut microbiome
This I agree.
and
That
diabetes
is,
association.
microorganism is inducing diabetes. I agree.
Yeah.
But whereas the first data you gave, correlative data, could be interpreted in both ways. That's why I asked the question.
Yeah. So then there are several studies in humans,
Okay.
and now
So what
we
is
can
that
talk
you
about
disagree with that? I mean,
today's scenario.
Yeah.
So I think a very important tenet in basic research is association does not imply
Yeah.
That I agree. I mean, I clarified that. But he's telling if I take the microorganisms from the normal first stage of the pregnancy lady and put that into the rats, they don't become obese and diabetic. Whereas if I take the microorganisms from the third stage where she's diabetic, the rats become diabetic. You can't question that, Chirag. Come on, give some freedom to scientists.
we definitely can question that, but
Yeah.
yes, so that data is, there's a lot of preclinical data which strongly goes in the favor of causation.
Yeah.
But we just do not have the data in human beings to suggest causation. So there's a lot of strong association data in that sense that, okay, the microbiome is disrupted in patients who are diabetic. Take be it type 2 diabetes, which is more of a metabolic condition, be it type 1 diabetes, which is more of an autoimmune condition. There are different mechanisms for both, and there is association with gut microbiome issues or problems, or dysbiosis as we call it, with type 2 as well as type 1, but different mechanisms. Now the thing is, what came first? There is no way to know that.
Yeah.
It's not like you take a patient who is about to become diabetic, as not yet diabetic. You've taken their microbiome sample. They got diabetes. Then again, you take their microbiome sample, and then you know it happened only because of that. Same thing with type 1 diabetes. So that those studies are not possible. We are not. It's not like we're doing them,
But what is the objection to this study?
So there, it would imply that if you were to give that person the right microbiome, or if you were to fix the microbiome, for example, then the diabetes would go away.
That may not be, though that may be questionable, but the implication of that microbes are causing diabetes.
I don't think it's that simple. There's a lot of different issues at play here. So it's not just that somebody's microbiome causes diabetes. There's a lot of genetic factors.
I agree.
Lifestyle factors.
I agree. But the rat is, common rat is, I mean, the rat that they have used it as a common genetic background, isn't it?
Yeah.
So what is the doctor's problem in accepting this scientific study?
We cannot use mechanistic data from rats and apply it towards human diseases.
I mean, I agree with your carefulness, but we can feel free that there is some conclusion
There is
we can.
definitely a link.
Okay,
I
great,
will
great.
agree to
No,
But let me.
No, I mean, see, even I would agree with him, because see, all these diseases, whether diabetes or CVD, they are very complex diseases, and you really cannot pinpoint a disease. And I always give argument in favor of microbiome. Today, if you look at even diabetes, if you do a search, you will find that even the human genome, you have 2 million human genomes of diabetic individuals are available in the public domain databases. But despite that, do we can we pinpoint at least 10 genes which are directly associated with diabetes?
We
No,
can't. Okay.
because these are very complex diseases.
That brings me to another
Yeah.
question. The question is to both of you. You can perhaps decide who would like to answer that. I am told that my diabetes is because of the genetic problem, probable genetic problem my father had, my mother had, and that's what the doctors have told me. You are telling that my diabetes could be also because of the organisms that
In addition
could
to
be.
genes. Yeah,
Okay.
yeah.
Now, is it possible to now separate this out? How much of it is because of my parental problems and how much of it is because of my microorganisms? Is it possible to separate them out?
It is not.
It is
It's
not
not
been.
possible.
Okay.
Absolutely.
But
It's multifactorial.
I
just
understand.
one
Yeah.
No, no,
Yeah.
please.
So it is multifactorial, and so it is not as simple as your father had it, you will have it. Your
I'm
lifestyle,
predisposed. They
you're
say
predisposed.
that's what they say.
So
Yeah.
let's say you have a baseline risk.
Okay.
So if your father or mother or nobody in your family had it, maybe your risk is 1. If
Okay.
your father or mother had it, maybe your risk is 1.1 or 1.2.
Okay.
You add a bad microbiome to that, maybe your risk
Yeah.
is 1.4,
Correct.
1.5.
Yeah.
You add no exercise to that, maybe your risk is 1.6, 1.7. You add a bad diet to that, maybe your risk is 2x.
Yeah.
So
Okay.
it works like that. It's multifactorial. There's a lot of different components. Your genetics and your
Yeah.
microbiome are 2 of those components.
I agree, but my interest in asking this question is, which among them is more dominant, genetic or microbial? We will also consider diet and all that.
I wish we had a simple answer.
So
to
we
that.
don't have the
We
answer.
do not have.
But you agree that both are important.
Yes.
Acha. To that extent, you are happy that
Yeah,
they are.
no, I'll just add to his answer and partially answering your question.
Yeah.
See, what the way as a microbiome researcher, what I strongly believe is that you are genetically predisposed for diabetes. What microbiome research can help you is that in addition to altering your lifestyle, I am confident that in future the research will lead to a stage that there could be some prebiotic or probiotic would be available after thorough research, by which if you are genetically disposed to get a diabetes at the age of 40 years, this can delay the onset of diabetes maybe forever, or maybe instead of 40 you will get it at 60 or 65.
Nothing wrong, I
that
think, to
this
be
science
hopeful,
will,
yeah.
or it could, and this has this has already been shown that it can reduce the dose of your medication. So if you are taking a certain amount of drug per day, that dosage can be reduced. And most importantly, if you he can add to this more, if you control the blood sugar levels and reduce the severity of symptoms for a longer period, it would reduce the diabetic related complications.
I mean, nothing wrong
yeah.
to be hopeful, isn't it?
Yeah.
I agree there is no data right now to prove exactly that this can be done, but nothing wrong to be hopeful, right?
There is,
because being
there's-
hopeful also promotes us to now look in the direction and do research.
There is data.
Okay.
So it's not like there's no data to point us in that direction.
Acha.
Microbiome manipulation,
Yeah.
as we can call it,
Uh-huh.
trying to convert a slightly less healthy or a dysfunctional microbiome to a more functional microbiome
Acha.
for that person. Again, I would like everyone to understand that everyone's microbiome is not the same. We're not trying to achieve the same microbiome for everyone.
Okay.
Every single person has a good microbiome for themselves.
Okay.
Right, and so it has been shown that microbiome manipulation through multiple different mechanisms, and maybe we can discuss them a little bit later, does help glycemic control, or basically how much you're able to control your sugars, can bring down your requirement for medication, can control your long-term complications and issues with diabetes.
Okay.
And that is just diabetes. That also extends to obesity.
Yeah.
Obesity complications decrease with microbiome manipulation. It extends to other cardiovascular diseases, heart disease, stroke, renal disease, or kidney disease.
Okay.
Beyond that, a multitude of other diseases
Okay.
as well.
Fine. I guess we have reached a stage where both of, or all the three of us are agreeing that it is good to now have a hope of using this whole science for medication. Okay.
Right.
We perhaps will now discuss on that further. For the time being, would you now both agree that there is enough science to now suggest that microbes are a inseparable part of our body? They seem to be now having more than our own cells in our body. They are operating in a way that we can't see them. They seem to be now affecting our diabetes, our obesity, our mental moods. I was told even Alzheimer's disease also is. So there are number of diseases and even our physiological disorders, etc. like sugar, diabetes and all that are also associated with the microorganisms. We can agree upon that, right?
Yes.
So if we agree upon that, then let us perhaps see later in another next episode on what is that being done by the clinical doctors or clinical field, and what is the state of this science in country, and can we now agree upon that?
Yes.
Absolutely.
Thank you. We'll meet in the next day. Thank you.